This is me and Cindy's phospholipid bilayer. Let's get started. So first up, for the phospholipid bilayer, we have the hydrophilic heads. Now the hydrophilic heads are made of cotton balls because hydrophilic means that it can absorb water. And since cotton balls imitate that exact thing, we decided to do that. Each hydrophilic head is made of a phosphate group and one glycerol molecule. Next up in the lipid bilayer, we have hydrophobic tails. Now we use pipe cleaners for this because they generally imitate the hydrophobic nature of tails, or water repelling. Each hydrophobic tail is a fatty acid, and there's two in each lipid. Next up, we have transport proteins. Now there are two different types in a cell membrane. There's channel proteins and carrier proteins. First one up is channel proteins. These are the red toilet paper rolls I used here. We use the red toilet paper rolls because they generally imitate the pathway that ions and molecules take when going through and from the intracellular and extracellular space. In channel proteins, this is where most diffusion takes place for small ions and water molecules, and one of these is osmosis, a process for higher to lower concentration through a partially permeable membrane, which is exactly what this is right here. We use the half toilet paper roll here simply because you could see the channel better. The other type of transport protein is carrier proteins. Now I'm going to take this out to show you how this works. Basically, a carrier protein binds to molecules, specifically larger and more complex molecules that can't get through with channel proteins, and they change shape in order to fit the molecule and send it across the membrane. Now, we use these Velcro straps because when a molecule comes, it'll come through the top here, and it'll open up and then close around to fit the molecule exactly. And same thing on this side. Molecule from inside comes and closes on in here. Now, each carrier protein is specific to a certain type of molecule or ion, so obviously not every single one's going to look exactly like this one. And all that is called facilitated diffusion. Next up on the bilayer, we have cholesterol. Now, cholesterol is important for the stability of the membrane, which is why we chose Legos. You can see it better on this side. Legos are typically known for building, so that's why we chose it for stability. Each one has its own hydrophobic head and hydrophobic tails, and it keeps the lipid bilayer from being too rigid in the cold or too fluid in warmth. Next up on the lipid bilayer, we have glycolipid. Now, it's kind of hard to see, but you can see that right there. That is hexagonal sugar residue. Now, we use glue gun droplets because it kind of makes the shape similar to what it actually looks like in real life. Now, a glycolipid is one of these sugar residues with a lipid. In general, they're receptor molecules, and they're similar to the next part of the bilayer. The next part of the bilayer all goes with cell signaling. There's the glycoprotein and the enzyme. Now, the glycoprotein is this big blue thing right here. It's one of these hexagonal sugar residues with a protein. Now, they act as receptor molecules similar to glycolipid, and they're involved in triggering chemical reactions within the cell and the cell membrane, which can also start up endocytosis or cell adhesion, etc. We made them the same color just so we could show you that it's all part of the same thing, and it helps with cell signaling later. Enzymes, the next part of the bilayer, also with cell signaling for glycoprotein. Now, the reason we use this red pom-pom here is because it's almost the exact same size according to ratio of the partially permeable membrane and the enzyme. Now, enzymes, at least here, are used to increase the signal of a signal in the cell that the glycoprotein finds. Now, the next part of the cell membrane is a process called cell signaling, which is important for the cell membrane because it gives the cell their appropriate response for external factors in the environment. Now, first thing that happens is a signaling molecule, or ligand, binds with the receptor up here. This triggers the glycoprotein to activate this enzyme. Now, this enzyme copies the response that the glycoprotein and receptor have, and transmits it and amplifies it all throughout the cell, which generates a response. Now, the reason the enzyme can cross over to the glycoprotein is because some signaling molecules are hydrophobic, so they can cross the fatty acid tails in order to bind. Now, the last two parts that we have in here are mainly just fun things. They're not needed in the cell membrane model, but they do help give it some variety. Now, right here, since it's kind of hard to see it, I'll take it out, is a globular protein. Now, a globular protein is interesting because it has a quaternary structure, which means it has many protein units or subunits in primary, secondary, or tertiary structures. This is why we had pipe cleaners folded in like this, because there's a lot of different shapes, and it can show all the different polypeptide chains, like looped helices or beta pleated sheets, so it's straight down the middle. For this model, it just acts as an extra integral protein. And finally, the alpha helix protein. Well, this is going to be really hard to see. But basically, it's this spring that goes all the way through, all the way to the top. Now, an alpha helix protein is also another integral protein that we added in here, but it's alpha glucose molecules connected in a helical pattern at hydrogen bonds in the amine group and glycosidic bonds at the ends. This is why we had a coil string, because it looks similar to the helix that it forms in real life. Thank you for watching my video of me and Cindy's bio project. Have a good day.